Okay, let's dive right into this explainer. Today, we're tracing the absolutely epic story of life's origins. We're going to zoom all the way in on the very first chemical reactions and then pan way out to the monumental leap into complex organisms. Basically, we're going to go on a journey from a sterile, lifeless rock floating in space to the vibrant, interconnected biological world we live in today.
To wrap our heads around this massive timeline, we've got a 5-part agenda for you. First, the primordial soup. Second, sparking the building blocks. Third, deep-sea batteries. Fourth, the multicellular leap. And finally, evolution's living snapshots.
All right, kicking things off with Section 1, the primordial soup, conceptualizing life's dawn. Long before we had modern labs, early scientists were already trying to frame this ultimate biological mystery. How exactly does non-living matter suddenly become alive?
We actually start our journey with a brilliant observation from Charles Darwin. Back in an 1871 letter to a colleague, he laid the philosophical groundwork for abiotic chemical synthesis. And what's wild is how he perfectly intuited the necessary ingredients. He talked about ammonia, salts, and an energy source like electricity. He famously called it a warm little pond, and honestly, that one phrase completely changed how we think about our origins.
That early pond idea sparked a whole century of incredible scientific pursuit. Tracing the timeline, we move from Darwin's theoretical musings to the 1920s, where Alexander Oparin and J.B.S. Haldane formalized the primordial soup hypothesis, which is basically the idea that a chemically reducing atmosphere could generate organic molecules. And then, boom, just a few decades later in 1952, we make the massive jump to physical lab simulations with Stanley Miller and Harold Urey.
Which brings us to Section 2, sparking the building blocks, the Miller-Urey experiment. Let's look at how those theories transformed into actual, testable science. So, the big challenge was, how do you actually recreate a primordial soup in a lab? To simulate the early Hadean Earth, Stanley Miller, working under Nobel laureate Harold Urey at the University of Chicago, had to answer one absolutely foundational question before he could even build his experiment. What exactly was in that ancient air?
Well, Miller settled on a very specific recipe. He took methane, ammonia, and hydrogen in a strict 2:1 ratio, mixed them with water vapor, and sealed the whole shebang inside a sterile glass flask. The idea was to mimic what scientists heavily thought at the time was a highly reducing atmosphere, essentially an environment primed and ready for organic synthesis, just waiting for a spark of energy.
And what happened next is honestly legendary. Miller applied a continuous electrical spark through those gases, simulating early Earth lightning. After just 1 week of this, the clear water in the flask turned deep red and turbid. By creating intermediate chemicals like hydrogen cyanide and formaldehyde, that spark kicked off what chemists call Strecker synthesis. And astoundingly, this successfully generated 5 distinct amino acids. They literally created the abiotic building blocks of proteins out of thin air. Well, simple gases.
But get this, the story doesn't end in 1952. Decades later, modern analyses of Miller's preserved vials by his former student Geoffrey Botta revealed even higher yields and a much greater diversity of amino acids than anyone originally thought. And here is why this is so profound. These exact chemical products remarkably mirror the organic compounds found inside the Murchison meteorite, which literally fell to Earth in 1969. So these chemical reactions aren't just happening in earthly glass flasks. They're occurring out in the cosmos, setting the stage for life across the entire solar system. How cool is that?
All right, moving on to Section 3, deep-sea batteries, a new model of abiogenesis. Let's see how modern geology gives us a major, cutting-edge update to that classic lightning in a primordial soup concept. So while the classic Miller-Urey model relied on sudden transient high-energy events, lightning and atmospheric sparks, modern research has actually shifted its focus toward the fuel cell model. This model looks at the continuous, steady, naturally occurring chemical energy provided by hydrothermal vents down on the deep ocean floor.
The absolute key here is that these hydrothermal vents act almost exactly like massive natural household batteries. They create highly specific pH and EH gradients, and it turns out life universally depends on these kinds of electrochemical gradients to drive metabolism. So using this battery or fuel cell analogy, researchers are now simulating prebiotic geoelectrochemical systems to figure out how these deep-sea energy gradients could have constantly powered the very first metabolic reactions. It's literally a continuous power supply for nascent life.
And this isn't just theory, it's a massive area of active collaborative research today.
Institutions like the universities of Leeds, Sheffield, and York are teaming up to investigate exactly this, testing what's known as the prebiotic fuel cell, or PFC model. Modern origin of life simulations are actually utilizing proton exchange membrane fuel cell components to test the energetic properties of these hydrothermal interfaces. We are essentially building ancient planetary seafloors right in the lab.
Okay, let's jump to Section 4, the multicellular leap, from chemistry to biology. Now that we've established our chemical building blocks and our continuous deep-sea energy sources, we need to zoom way out to observe the monumental leap from single-celled life to complex, interacting organisms.
Now, you might be thinking multicellularity is just a bunch of cells sticking together, but it's so much more than that. While simple cellular aggregation is a good start, true complex multicellularity, which kicked off over 600 million years ago for animals, requires distinct, highly sophisticated behavior. We're talking about intricate cell-to-cell interconnection, communication, cooperation, and cellular differentiation.
Think of this transition almost like a business. Originally, single-celled organisms were acting as independent workers. But eventually, evolutionary pressure made them realize they could survive a lot better by forming a corporation. They chose altruistic cooperation over individual self-interest. This little micro-narrative is completely crucial to understanding our own biology. These organisms had to literally sacrifice their individual reproductive rights and divide up the labor, all for the synergistic survival of the greater biological entity. Which leads us to our final part, Section 5, Evolution's Living Snapshots, Modeling the Transition. How do we actually study a biological shift that happened hundreds of millions of years ago?
Well, scientists don't just rely on ancient fossils. They observe living, facultative multicellular organisms that still blur the line between solo and group living today. Take the prokaryote Myxococcus xanthus, for example. When starvation or predation creates adverse environmental conditions, these genetically heterogeneous single cells are literally forced to rapidly group together. And once they aggregate, they begin to differentiate and divide up the labor just to survive the harsh environment. It's an incredible real-time display of the leap to multicellularity.
This is exactly why scientists utilize a diverse array of these living snapshots. You have things like Dictyostelium slime molds, where cells altruistically differentiate into spores based simply on their genetic ploidy status. You have the single-celled paramecium, which demonstrates epigenetic inheritance, meaning they can tune their somatic development to environmental stressors like heat shocks and pass that adaptation on without even changing their DNA. And of course, yeast, which is actively used in experimental evolution to explore how cells first began adhering to one another. By studying these specific organisms, scientists are capturing a real-time snapshot of the regulatory changes and developmental plasticity that bridge the massive gap from the microbial world to the animal kingdom.
Which brings us to a massive question to ponder as we wrap up. If we can reconstruct this epic journey, from simple atmospheric gases sparked into amino acids, to deep-sea vents powering early metabolisms, all the way to single cells sacrificing their independence to build complex bodies, how might these planetary simulations guide our search for life on other worlds? I'll leave you with that provocative thought to consider, because understanding the rigorous, step-by-step blueprint of our own origins provides the exact lens we need to explore the rest of the universe. Thanks for joining me on this explainer, and keep questioning the world around you.